Okay, so welcome back. This is episode or video 4, part 4 of this lecture series. Probably be the final part of this lecture series. Before we get going, though, I don't know if any of you are paying attention much to the presidential debate or anything, but it's getting really interesting. They just had their first presidential debate about like a week ago, and they're gonna have the second one on Sunday. This presidential debate is super. This whole election is really important. The stakes are high. It's like between two people. So I just like to watch my favorite clip or my short, nice recap of the episode of the presidential debate.
Welcome back, everyone. There's still more surprises in store, but right now the contestants are playing a round of I Can Do This. And go.
Well, I can do this.
Woo-ha! Woo-ha!
Well, yeah, I can do this.
Everybody sing.
I doubt you're quite that good.
And I assume I can ask you to sing, Mr. Trump.
Okay, I don't want no big, fat, ugly baby. I want a baby on fleek.
That's really nasty to squash a baby's self-confidence like that.
Hey, have to say, you suck. Well, that's just stupid. That is stupid.
We know it.
Okay, so see, the presidential debate is fun. Voting is fun. Make sure you go out and vote. That's important. We need lots of people voting this time. Anyways, so just a quick recap of what we said last class. You have to be able to do a pedigree. I guarantee you there'll be a pedigree on the lecture exam. I guarantee you there'll be a pedigree on the practical. You want to make sure you can do it. That's very, 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 very important. So make sure you're able to do that. That's very, very important that you can do that. So, Mendel did all these different experiments, but then there's someone else who's really important named Thomas Morgan. He did some additional experiments and found out some more information, but he used something called Drosophila melanogaster. That's another way to say fruit fly, the common fruit fly. He did some experiments on them, and he mated male flies with white eyes, these are mutants, with females with red eyes. So he mated white-eyed mutant males with female red-eyed wild types. Which one is more common? Red eyes, because they're considered wild type. So he mated these white-eyed males with these red-eyed females, mated them. All of the offspring were red eyes. Well, that sounds a lot to me like what Mendel found. Then he mated all these red eyes individuals, but he found something weird. In the F2 generation, there was a 3-to-1 ratio, which is normal, of red eye to white eye. But for some reason, only males had white eyes. Every time he did this, well, for some reason, females never had white eyes when he did this experiment, which is a little strange. So because of this, he said, maybe it's just not as easy as we've been talking about. He believed that that white-eyed mutant allele is found on the X chromosome. So because of that, this supports that now chromosomes and DNA is being inherited. And it's kind of interesting that all of these males had white eyes. Why? Because they had this mutant allele. So we'll take a look at this. So here's a red eye wild type and a white eye mutant. Thomas Morgan then mated red-eyed females with white-eyed males. All of the offspring, male and females, had red eyes. Then he mated them with each other. All females had red eyes. No females could have white eyes. Half of the males had red eyes, half the males had white eyes. This is weird, but it shows that the white eye allele, the white eye mutant gene, was found on the X chromosome. Females have 2 X's, but both of their X's did not have the white. One had a red, one had a white. Males, how many X's do they have? Just 1, right? So this X was the white eye, this one was red. And we'll talk about that. If that doesn't make sense, we'll do a couple explanations. So here's the female, and there's the male. If he was going to make a male here, he had to pass on the he had to pass on the Y, which doesn't even have the eye color gene on it. So he passed on the Y. Then this individual had to have gotten the red eye from the female. So we give it a plus because it's the wild type. So this individual is a male with red eyes. This individual, the females, they got a white eye allele from dad and then a red eye allele from mom, but they still show red eye because red eye would be dominant. Then these had offspring, these male and females, and then if they made a male, the dad gave the Y. So the mother gave, in this case, a red eye. So this male had red eyes. In this case, the mother gave a white eye. So this male had white eyes. So here, if you look, there's no homozygous dominant, homozygous recessive. There's no heterozygous. They just have one. You either have 1X or 1X. They have this, they're red eye. If they have this, they're white eye. These, the same thing happened here, but except they have 2 X's. So this individual, they don't have white eyes because they don't have 2 white eye alleles. This one, they have white eyes, even though they only have 1. That's weird, but that's because they have a Y chromosome that the dad gave. So hopefully this makes sense. If not, I'll take a look at it, pause and rewind. Hopefully this makes sense. If not, I'll describe it, I think, in the next couple slides. So remember, humans and some other animals, we have chromosomes which determine the sex of an individual. There are 2 sex chromosomes. Remember, there's an X and there's a Y. Here's the X, and then there's the Y. X and Y. X is really big and often has different genes on it. Y is often small. And remember, if you have a Y, you're a guy. Human males are often XY. Human females are often XX. They're not males because they don't have a Y. The presence of a Y makes you a guy. I said that again. That's like the bajillionth time I've said it. So here you can see that I know this is a male because they have a Y. I know this is a female because they have an X. Whenever the male makes an offspring, half of his sperm are going to have an X, half of his sperm are going to have a Y. If he makes a male, well, then they only got 1 X chromosome, and they only got that from mom. They couldn't have gotten an X from dad because, well, dad gave them a Y. So the reason that a Y makes you a guy is because on the Y chromosome, there's a gene called the SRY gene, the sex-determining region on the Y. This gene promotes the development of the testes while you're just an embryo. During your fetal development, before that, in your embryonic development, this gene says, I'm a guy. So you start developing guy stuff, like testes. The X chromosome is really big, remember? You can see it here. It's a lot bigger than the Y. On it are many genes that don't have to do with sex. They don't have to do with male or female. They have to do with, in this case, eye color for flies. Not really for humans, but eye color. And it's different for humans. We have genes on there like for hemophilia, like a liver blood clotting disease, and color blindness and so forth. So the SRY gene, that's now why we know. We're being more specific. This is why if you have a Y, you're a guy, because you have this SRY gene on your Y. So a gene could be sex-linked. That means it's found on a sex chromosome. It could be found on the Y chromosome, so we call it a Y-linked gene. Doesn't really happen. Most are X-linked genes. There are a lot of them because the X chromosome is big. There's not a Y-linked gene. A lot of them because, well, the Y is so small. There's not really many of them. So that's important. Some genes are actually found on these sex chromosomes. So X-linked genes, they follow a specific pattern of inheritance. If it's recessive, generally for you to have a recessively inherited condition, remember, you'd have to have 2 copies of it. Because if you had one dominant, you wouldn't display that recessive condition. So a female, she has 2 X's. So a female, in order for her to express this disorder, let's say like white eyes, she would have to have 2 copies of that X allele. She'd have to be homozygous for it. But a male, since they only have 1 X, and they have a 1 Y, and the Y makes them a guy, they only have 1 X. It doesn't matter. There's no homozygous and heterozygous, like I said. We use a term called hemizygous. That means you only have 1 copy. Males only have 1 X chromosome. Whatever's on there, it's what they are. There's no dominant recessive. It doesn't matter. Whatever's on there, that's what they are. So this is often seen in men's. In men, hemizygous is really only seen in men because they have 1 Y, which doesn't have the gene, but then they have an X. which carries that gene. So it's really important to understand what is hemizygous and how does that happen. Here's a picture of that. This fly here is hemizygous for red eyes. This fly is hemizygous for white eye. So because of this, even though this female has a white eye allele, the mutant allele, she doesn't express it because she has a dominant. Males, it doesn't work like that because they only have one copy. Whatever copy they have, they got. Because of this reason, we see this trend. X-linked recessive disorders are a lot more common in males than females. Because if you think about it, a male only needs one copy of that disorder because they're hemizygous. Females, it wouldn't be as common because they'd have to have 2 copies of that recessive gene. It's not very common for females to have white eyes because, well, they'd have to have 2 copies. Males, they only need 1 copy, so it's more common for a male to have that white eye than the female. That's because of the way inheritance works. So, X-linked inherited disorders are seen in humans. Color blindness, hemophilia, Duchenne's muscular dystrophy. These are some different examples of X-linked disorders. Color blindness is the most common one. So color blindness is an X-linked recessive disorder. What that means is the gene for color blindness is found on the X chromosome. If you are recessive for it, if you have 2 recessive or a recessive allele for it, then that means that you will be colorblind. So for a male to be colorblind, he only needs 1 copy of it since he only has 1 X. For a female to be colorblind, they have to have 2 copies of it since they have 2 X's. The most common type is red-green colorblindness. X-linked recessive disorders are more common in men than females. 8% of all men have some form of colorblindness. That means in our class, probably 1 or 2 of you have colorblindness. 1 or 2 of you are partially colorblind. In every class I've taught, there's always someone in there that's colorblind. It's probably the case with our class. But the chances are, most likely there's not a woman in our class who is colorblind. It's kind of crazy to think about. There's likely a colorblind person in our class. So you can detect colorblindness by this. So you might find out right now, oh, I have some form of colorblindness. What do you see? What number do you see? Right? What number do you see here? Do you see 71? Do you see 74? What number do you see? So if you see 71, then or like maybe 74, then you don't have a form of color blindness, probably, at least red-green in this case. If you see 21, then they say that I believe that's what they say, is that you have some form of color blindness there. So I think if you see 71 or 74, you're good. You don't have this form of color blindness, but if you see a 21 here, then you probably have some form of color blindness, at least red-green in this case, and there's lots of other ones. So quick question, what happens if you see this, right? If you see this, what kind of condition do you have? Well, if you see this and that's all you see, you're blind, right? So if you're blind, you see this. If you're colorblind at this, I believe you see a 21 in this case. And if you're not colorblind in this red-green spectrum, then you probably see a 71 is what you'll likely see, possibly a 74. Or maybe if you're colorblind, you see a 24 on this, not 21. But if you don't see a 74 or 71, you see a 20-something, then I think that means you have some form of colorblind. But go see a doctor about that. I'm not here to diagnose you, but it's kind of interesting to think about. So the reason this happens, because a male can only give an X or a Y. And if he's colorblind, this male here is colorblind. If he has a boy, he passes on the Y, so he can't pass on his colorblind allele. If he has a girl, then he passes on, he's guaranteed to pass on his colorblind allele. These 2 girls are carriers for the colorblind allele. Mother only can pass on capital N in this case, so all of her males are normal vision. But in this case, if a female is a carrier, dad passes on Y to all the males, there's a 50% chance that they're going to have an offspring that has colorblind because of this. So males always pass on a Y to their son and always pass on an X, and then the females do that. So kind of make sure if you do a Punnett square that involves a sex-linked gene, make sure you know that male, if you're making a male, they pass on the Y, if they're and then so forth. So I want you to complete a Punnett square for a cross between a normal male and a female carrier. So go ahead and complete a Punnett square right now. Pause the video and complete a Punnett square for a male who's normal and a female who's a carrier. So first thing you got to think about, what are the genotypes? If a male's normal, what would he have? If a female is a carrier, what would he have? She have? So pause the video and try to figure it out and draw it on your own. So draw the Punnett square. If a male is normal, what would his genotype be? He's XY, but if he's normal, he'd have to be XMY, right? A female, if she's a carrier, what does that mean? Does she have a Y? No, she's not a guy. We put XM, X_m. Then drop down the stuff. Now we'll find that if these people have a child, well, half of their males will be colorblind. Half of their males will not be colorblind. These males are hemizygous for this. Hemizygous for normal, hemizygous for colorblind. This individual is a female who is not colorblind. She has normal vision. This individual is a female who is not colorblind, but she's a carrier. So you want to make sure you can do a sex-linked Punnett square. That's extremely important for genetics. Finally, females have 2 X chromosomes, right? Here's the interesting thing. In females, one of those X chromosomes is actually inactivated. You have 2, but one of them randomly turns off. So X inactivation is when in female mammals you have 2 X chromosomes. But during embryonic development, when you're just developing as a pre-baby before you were even born, really early on, like in your first 1, 2, 3 weeks, half of the X in your cells turn off, half of the X in your other cells turn off. So they're randomly inactivated. So when you were just a 16-celled girl, it then flipped a coin and said, if heads, this one turns off. If this one, it turns off. So only in females, one would randomly turn off and the other one would randomly turn off. So it's a random inactivation. So like I said, if you're a 16-celled female, then cell 1 is going to flip a coin and be like, oh, this X from dad turns off. Up, flip this coin. Oh, this X from mom turns off in this cell. So that's what happens. X inactivation, they randomly turn off. They randomly become inactivated. They become something called a bar body. The X chromosome becomes randomly inactivated. So if a female is heterozygous, if she has this, one of these are going to randomly turn off. This one might turn off in the liver, or this one might turn off in the liver. Color blindness might turn off in the eye. Good, she's not colorblind. Or this one might turn off in the eye, and then she'll still be colorblind. It's kind of weird to think about. X inactivation happens. It becomes a bar body. So if they're heterozygous, they might become mosaic for that character. And I'll show you a picture. Here's a calico cat, right? So this is a female. And if you look at this, during embryonic development, they have an allele for orange fur and an allele for black fur. Only this displays complete dominance, right? If orange is present, they become. If orange is present, then orange might be. I believe black is dominant, and they'll become black. But they might randomly turn off black here. It might become inactive and become a bar body, and so so the orange is actually able to be displayed and be expressed. So here in this spot where there's black fur, that's because the orange turned off. Here in this orange fur, that's because the black turned off. So that's why they actually appear like a mosaic. Why couldn't a male be a mosaic? Why can't you have a male calico cat? X inactivation doesn't happen in males. Imagine if it did. Look at this male. If you turned off one of his X's, he wouldn't even have an X. And that would result in spontaneous abortion or a miscarriage. That can't happen, right? So one of the X's is going to only turn off in females and becomes a bar body. Males, that doesn't happen. So that's a picture of that. So here's a nice review sheet that kind of tells you what is complete dominance, what is incomplete, what is codominance, blah blah blah, what is epistasis, what is polygenic inheritance, and so forth. So now we'll briefly get into this. We'll do a small portion of this, and then we'll cut it close, or we'll cut it off, and then we will pick up next class. So now we're going to get into the structure and function of DNA. What is DNA again? How does it work? What is RNA? We're going to talk about DNA replication, how DNA gets replicated, and then how we make proteins from that. So if you remember, DNA is the master blueprint of your body, right? It contains the hereditary genetic information, and then that directs the proteins and how they develop inside of you. The DNA contains your genotype, and that determines your phenotype and how you look and everything inside of you and how you act. Because we have chromosomes, condensed units of DNA and protein. DNA wrapped around a histone protein called a nucleosome. They knew that DNA was made of they knew that chromosomes were made up of DNA and protein, but originally they didn't know if DNA was the genetic material or if protein were the genetic material. Thomas Morgan proved that chromosomes had the genetic information, but we don't know if it was DNA or protein because chromosomes are made of DNA and protein. So we did a science experiment, or at least Frederick Griffith did in 1928. He had two different strains of bacteria. One was bad and caused a disease. One was harmless and did not cause a disease. He killed the harmful one. He killed the harmful bacteria and put the dead carcass of all these dead, harmful bacteria in the test tube with the living, healthy, harmless cells. He did that, and look, he had these. If you have this pathogenic cell, put it in a mouse, you kill it. It's harmful. If you have this non-pathogenic cell, the healthy cell, then the mouse doesn't get affected. If you kill the healthy, or if you kill the pathogenic disease-causing cell with bacteria, with heat, then they live. But if you kill it with heat, put the dead carcasses in the test tube and mix it around, the cell dies. Somehow, genetic information was exchanged between the dead one and then the living non-pathogenic one, and that living non-pathogenic one became deadly. Why? There's something called transformation. The genotype changed. The phenotype changed because foreign DNA was incorporated. This dead carcass of the disease-causing, the DNA was passed on to these harmless cells, and they became harmful. So that's an example of transformation. You can kind of, hopefully you need to be able to understand that experiment. So around 20 years later, they eventually found out DNA was the genetic material. Many people didn't believe them because they didn't know a lot about DNA. They thought it was so small it didn't matter. They actually thought it was protein that did the genetic information. But later they found out it wasn't. We now know nucleic acids are the big macromolecules made up of nucleotides, the individual monomers, the individual units. There's DNA and RNA. Nucleotides, like we said, are made up of a nitrogenous base, G, C, A, T, or U, a pentose sugar, either DNA or RNA, and they have a phosphate group, is what we've said. Here's a picture of that: nitrogenous base, sugar, pentose sugar, and then a phosphate group. That's what nucleotides look like. There are 5 nucleotides you need to know, and we already knew about these. There's UGCAT, uracil, guanine, cytosine, adenine, thymine. Uracil is only in RNA, thymine's only in DNA. Then you need to know how they bind, right? And we'll talk about that in the next couple of slides. So after they found out DNA was a genetic material, they then need to figure out what does it look like, and then how does it work. So there's this awesome woman named Rosalind Franklin. She did a technique, and she was the first one able to look at DNA. So she used this technique called X-ray crystallography, and she did it, and here's that picture she took of DNA. And she saw, and then from this, they determined, with Watson and Crick, they found out the DNA has this double helix. James Watson and Francis Crick, later on, Watson and Crick, they determined DNA is a double helix and DNA and how it exists. So here's a picture of how they did double helical structure of DNA, kind of stolen from the work of this woman, Rosalind Franklin. So they discovered DNA was a double helix, a spiral staircase, and it was anti-parallel, right? Remember, one strand runs from top to bottom, 5 to 3, and the other one runs from 3 to 5. They're flip-flopped. The same exact strand, but they're flip-flopped. So here's how that is. If one strand goes CGG, blah, 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 then the other strand is going to be the exact opposite and then going the other way. So that's just how DNA works. So, because of this, Watson and Crick finally found that there's complementary base pairing. G always base pairs with C. A always base pairs with T. And in RNA, A always base pairs to U. That's what they found, complementary base pairing. This is how it always happens. GCAT. G always pairs with C. A always pairs with T in DNA. A always pairs with U in RNA. So because of that, you can do these percentages like we've talked about. G always pairs with C. A always pairs with T. So from this, you can do this, answer this question. If 30% is adenine, what percent is thymine? Well, it has to be 30%. And then what percent is uracil in DNA? 0. And then from that, if 60% are A and T, that means 40% are G and C. Divide that by 2, and 20% and 20%, like we've already found. So because of this, this is where we're gonna, what we found is we now know what DNA is. Since the strands are complementary, the strand can actually act as a template to build new strands off of. So that can actually happen. That's called DNA replication, where we use one DNA strand and we use it as a template to build another strand off of it. So if we have this, A, C, T, A, G, what does the other strand have to be? What would be here? What does A always bind to? T. What does C always bind to? G. A. T. C. That's how it always happens. If you know one side, you always know the other. DNA is double-stranded, and if you just take off one strand, you could write in the other strand from scratch. So you want to make sure you know how to do that. That's extremely important. So this is what happens. If you have a parent DNA molecule, it looks like this. During DNA replication, they split into 2 templates, and then we make a new strand to make for DNA replication. So we use each strand as a template, and then we replace the other side. And we can make exact copies based off this. Now, we didn't know how DNA replicated. We knew it did. We didn't know what form did it replicate. We didn't know if it was conservative, right? Where you have one strand and then we make a whole other strand based off of it, and they replicate. We didn't know if it was that, or if it was semi-conservative, or we split apart the strands and then we make a copy based off that, or if it was dispersive, where we looked at it, cut up a bunch of pieces, and this is like a candy cane, brand new, duplicated, and so forth. We didn't know. We did experiments. We found it wasn't these. It's semi-conservative. We found out that when DNA replicates, the 2 DNA strands separate, and then they consist of one strand that's old, one strand that's new, just like this. When you have a parent strand, they separate, and then we use these as a template to build their complementary strand. We separate this, and something goes and puts in the complementary strand. That's DNA replication, and it uses a semi-conservative mechanism to do that. So that's extremely important. That's how it works. So make sure you're able to understand that, and so forth. So now this is where we'll end the online lecture series. This is the end of part 4. So make sure you have watched all 4 of them. That's really important, and you'll have a quiz on this material. And we're going to pick up around here next in class. In class, we're going to do a practice practice pedigree, and then we'll pick up here. And if we have some time, we'll go back and do a genetics review. But we will not have time to do the Jeopardy. Although I will upload Jeopardy, we're not going to have a chance to do that in class. So have a good weekend. Stay safe. Have a great week. Make sure you look over this material. There's quite a bit of material, but in a review, some of the more difficult things we've seen, make sure you're able to do a pedigree. You have to be able to do a pedigree. Make sure you're able to do a Punnett square, and even a sex-linked Punnett square. You don't have to do a sex-linked pedigree, but you need to be able to do a sex-linked Punnett square and a regular Punnett square, and make sure you can do pedigrees. That's very important. Make sure you can do it for a condition that's autosomal dominant, where it's dominantly inherited, right? Where just one copy and you have it, like we said for achondroplasia. Or make sure you can do one where it's recessively inherited, like cystic fibrosis and albinism. So make sure you're able to do that. That's very, 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 very important. A good portion of your exam will be on that. So make sure you can do that. That's it. Be safe.